talking to you. This is Consultant Biology, and it's the last day of school before vacation for 1825. And I'm Mrs. Bolvin, and since I can't be with you today, lucky you, you're still going to be able to hear my melodic voice give you some notes today. So let's get to it. So today we're talking about translation, and translation is making a protein using the directions read from an mRNA. And the last time we were in class, we talked about how the codons on mRNA are little three-letter words that code for a protein. And now this is the process that's putting all of those little mRNA words together to create a protein, a string of amino acids. So translation happens at the ribosome. And you need that other RNA that we haven't talked about that much yet is tRNA. And that's responsible for bringing the correct amino acid to the ribosome so it can be attached. So the tRNA, just like the mRNA had a codon, the tRNA has what we call an anticodon. And you're like, what the heck is that, Bolvin? So anticodon is 3 tRNA bases that are opposite the mRNA base. And as you can see down here, on the mRNA, if we have a U, the tRNA is going to have an A. So you're kind of going backwards here. The C on the mRNA means that there's going to be a G on the anticodon. If there's a C on the mRNA, there's going to be a G on the anticodon. So these 3 bases are kind of like it ensures that they're going to actually fit in the right place on the mRNA. Because if this were a C here on the anticodon instead of an A, it wouldn't be able to fit into this place that the codon has it wouldn't fit. The pieces wouldn't fit together. It's like a puzzle. So if the mRNA codon is UCC, as it is in this picture, the anticodon bases would be AGG. Now, that becomes important because that ensures that the right amino acid is being brought in there so that it can attach to the protein. If it didn't have these bases in the anticodon, it wouldn't fit and it wouldn't be able to be attached. Now, at the other end of this thing is the amino acid. It kind of looks like a big old clover leaf, and this is one end of the clover, and on the stem end is where the amino acid is attached. So these are the steps in translation. So first, to start a protein, you have to have the AUG on the mRNA slides into the first slot. Ribosome kind of looks like a hamburger bun, but it has 3 grooves in it. The first slot is where the mRNA AUG, it fits in there, the ribosome comes together, and then we need to get the tRNA with the corresponding anticodon, which would be UAC, and that has methionine attached to the other end. Remember last time I told you that methionine starts all the proteins, and then sometimes it gets snipped off and sometimes it doesn't. The ribosome has to know where to start, and this is the signal to start. So it brings the tRNA brings the methionine amino acid to it. Then the UAC anticodon bonds to the AUG codon or mRNA. So they kind of come in here. This is like a picture a little bit after, we're a little bit further along in the process. So the first tRNA, once that's bound in there, the first tRNA slides over with its mRNA. And then the next tRNA brings the correct amino acid, and then it binds to the mRNA. The 2 amino acids, as you can see in the picture here, hook up together, and it just keeps doing this process. The amino acids attach to each other, slide over. It's going to continue to do this, and every time it gets to the 3rd position over here, whatever tRNA was there, now its amino acid is stuck to the one before it, and it gets kicked out and it can go and find another amino acid to pick up. So when the next tRNA arrives, it kicks out the first one, leaving the amino acid attached. Keeps going until it reaches a stop codon, and the stop codon says, "Stop right there!" And then the ribosome comes apart, and voila, we have a protein. So 7 easy steps to getting a protein. So the ribosome opens, actually 8 steps, sorry about that. 8 steps. The ribosome opens and the mRNA is released, and we have a protein. So just to recap a little bit here, the central dogma, or the big idea in science, is DNA to RNA to protein. So DNA, it hangs out in the nucleus, never leaves the nucleus. Transcription happens, and we get some mRNA. The mRNA then goes to the ribosome, and the ribosome carries on translation. So transcription makes mRNA, translation makes a protein. During translation, the tRNA brings the amino acids and attaches them to make a protein. Okay, well, that's it for this time. Have a fantastic vacation, and I'll see everybody when they get back. Ms. Blanchette will let you know what you have to do next, but have a great vacation. See ya.